How are you doing?
Okay. Hi, Bradley.
Hi, how are you?
Oh,
So, yeah, how's everyone doing?
okay. I'm confounded.
Yeah, we had
I'm
a good meeting on the breast cancer physics earlier. Went over some things. Yeah.
oh, breast cancer physics?
Yeah.
Really?
Yeah. There's a group that's we're looking at sort of imaging breast cancer and different ways of doing that. So.
Okay, good. Need to get rid of the squisher.
Need to get rid of it. We're using it.
You need to get rid of the squisher.
The squisher, huh? How much does it hurt?
Lots.
A lot, huh? When they say does it hurt you, then they squeeze it more, huh?
It's got to. It was my comment on the whole thing was it was designed by a mechanical engineer.
Okay.
Yeah. Mechanical engineers are usually arrogant and like to do things mechanically perfectly, which doesn't always jive with soft biological parts.
There is a logic behind it.
Yeah, I know, but that doesn't. That means that I know several people who will not go get a breast exam.
Oh, they will not. Because of that.
Absolutely.
Okay.
Absolutely refuse.
Absolutely refuse. Okay.
Yeah.
The logic is the thinner the tissue, the easier it is to image it.
Well, whatever. They're not going near the damn thing. And I had someone, a friend, she's now deceased. She had breast cancer. And she went in one time to have an exam. And she said there was something wrong with the technician that day. She didn't care whether the whole thing is flattened permanently. Like, it was a bad experience. And this is a person who went regularly to get this done because she had breast cancer.
Okay.
Yeah.
Okay. Well, we're trying to devise a device that finds small breast tumors and then destroys them. So a woman goes in, gets diagnosed, and then immediately cured.
Yeah. Well, that's fine, but if you could find an initial thing that did the diagnosis that wasn't so painful.
The idea is to do it in one machine all at once. There's no initial. Okay.
You can use that as a goal, but it won't work.
Yeah, it won't work. There, Bradley. The voice of experience.
You need the diagnosis first, and then if you say you're going to zap them, then they'll think about it, and then they'll allow you to zap them, or they won't. But what I'm saying is your zapper is a really good idea as the second line of defense. The first line of defense is go image it. If you're going to image and zap, people are going to go, no, even worse than what they're doing now.
You think so, huh?
Oh, I know so. They
what if they're offering
in the area that I'm in, they're going to pray it away, you know. I'm sorry, it's just the way this area is.
Okay.
I've seen them on the mask mandates and the COVID vaccine. It's like, oh, come on, guys.
Okay, so the problem is, Bradley, the problem is twofold. One is having the breast in the same position during diagnosis and treatment.
Yeah.
Okay, and I've always said you can't put a breast in a machine the same way twice.
Yeah.
No, you can't.
So that's why we want to put it in once. What Susan's concerned about is mostly the pain, right?
Right.
Yeah. Well, and you think that maybe if the technician pushes the wrong button the wrong way, the whole thing is going to
Oh
go.
God.
You're gonna lose some bits.
Yeah. Oh well. You could have the patient control the button. I had that once, but not for breast. It was for some sort of opioid, to painkiller, where you could control the painkiller.
Oh.
Okay, and this came I guess in the last 10, 20 years it came in, but it allows a patient to give the minimum dose that kills the pain, rather than somebody deciding in advance what the pain dose is. So they control the injection of the painkiller. Okay.
Well, that would be good. So along with this,
Maybe we should have
you're going to have morphine, are you?
Yeah, maybe there's offer morphine. Maybe people will go for breast protection then.
Yeah.
Yeah, maybe. Anyway, there are two problems with breast compression. One is minimizing the pathway through the X-rays, and the other is the less you compress, the more jiggly you got it. So the less precise you are. So we might need to get a mechanical engineer involved, preferably a female who understands the problem.
Yeah, female mechanical engineer. That sounds better.
Okay. Okay. I take your warning. We were just going with a standard breast compression, which is, believe it or not, 3 to 5 centimeters. I think we'll continue with it, and but take your warning, because we do have to look at some literature on why compression has to be 3 to 5 centimeters. Is that due to the X-rays, or is it due to? Well, it can't be due to mechanical positioning, because if they find something, the woman has to come back for treatment. Treatment is now done separately. Okay. Yeah. Our suggestion is to do it all at once. Okay. And the point is to avoid the repositioning problem, because the repositioning problem will involve a three-dimensional transformation, which would have to be determined in each case individually. Okay, and we're aiming for tumors that are really small, 2 to 4 millimeters. Excuse me, 2 to 4 millimeters. And so positioning is very important.
Yeah.
Okay.
My sister discovered her breast cancer by herself as a large lump.
Oh. Yeah. Well, we're trying to catch it so early that it's curable.
Yeah, instead of what she went through.
But I worked with 3 sets of epidemiologists, and we predicted that by extrapolating clinical data, that if you can catch a tumor at the 2 to 4 millimeter diameter, then you'll achieve greater than 99% cure rate. Okay, so that's why the positioning problems are so important, because it's such high resolution. I mean, you have to understand, standard computed tomography done on a body has a resolution. I think the best resolution is about 0.5 millimeter now. Okay, and that's so. And the problem with repositioning is, okay, what do you reposition to? You got to find the same tumor again. Okay. So, okay, Bradley, we should let you do your thing.
Oh yeah. I thought Susan. I don't know if you gave an update or not. I think maybe that got.
No, me, I'm still fighting with console and mice. So I figured out that I've got to make a nice little hexagon to surround it instead of cylinder, because the cylinder has 4 nodes to describe it. So I'm going to make this hexagonal thing with lots of nodes around the side, and then I can hook up my tissue to whatever one. So I'm making, I think I'll make it out of, I don't know, some interesting metal or glass or something. Although I could make it a beef ring, apparently.
Oh, okay.
They have beef in as a substance in the materials library in Comsmol. So I'm going, well, should it be glass? Should it be titanium? Should it be beef? Does it matter?
What are you making?
My tissue collapses because I don't have the half twist on the cells. So I'm making a ring around it as though it was attached to the rest of the tissue in the body. So I have.
Wait, what collapses? Cells. I missed something. What collapses?
Well, the matrices for the tensegrity that I'm making has negative eigenvalues or something, or empty matrices, because I have two things attached to one node, 2 or 3 elastics attached to one node, or one rod and 2 elastics, so it doesn't like it. So I'm suspending it like it was a 3D spider web.
Okay.
Because that's actually what our epithelial tissues look like.
Oh.
Kind of a 3D spider web. Like they're all attached at the top for sure, and then they're attached at the bottom to the basic tissues. They're not really attached at the sides. Like that's.
They aren't.
They are.
Really? Except right at the 2 ends. That's interesting.
Well, if you can find, if you run across some literature saying where all these cells are attached to each other, it would be great. But I'm reading that they're attached at the top and the bottom. Like,
interesting. Okay, the junctions. Yeah.
Yeah. Yes.
Okay.
And thanks to the literature you said, it does form a ring of.
Let me tell you one strange observation I made. I once used the high voltage electron microscope in Albany, New York, on axolotl epithelium. Okay? It was probably neuroepithelium. And the pictures, you're dealing with thick tissue, so you don't get sharp tissue. You don't get sharp images. But it looked like microtubules went right across from one cell to the next.
Yeah.
Which of course they can't, but maybe they hit a junction and maybe you get microtubules on the other side.
Yeah, they just continued on. Like microtubule joint.
It was an impression. Pictures were not convincing.
Yeah, well, I was going to have it so that you could mess with the microtubules and see what that did to the cells.
Okay.
Spiderweb thing.
Okay. Well, we still have the outstanding question as to what are the stability properties of the cell state splitter.
Yeah, well,
which I think is a tensegrity structure.
Well, I have to get a working model first.
Okay.
Think about other things, but it's just it's it's being a pain. I'm learning COMSOL and I think I gotta do this. And I was told by my advisors that couldn't I just get something or other and throw it together and get it to work. And well, no, I actually have to learn COMSOL and rather some sort of depth.
Yeah, advisors don't do that.
Well, it was just supposed to be an initial thing. Can you get it to work? Well, it's a tensegrity object, so.
Okay. I have a general question. If you have a connection matrix for tensegrity parts, can you tell whether or not it will collapse or stay three-dimensional? Is there a way of doing that?
I have a paper there where they used some continuum mechanics on some tensegrities.
Yeah.
And they found areas where it was unstable and areas where it was not, and actually managed to get some interesting materials.
Oh, okay. Could you send me that paper, please?
Yeah. That was I'm hoping to use that actually as my main method of modeling, because it looks like I could just put it in MATLAB and just use continuum mechanics and stop messing around with things that give me negative eigenvectors, and then they don't give me the matrices so I can see where it goes. So I'm doing guess by gosh, and you don't do that. You don't do that.
Okay. Well, yeah, Bradley and I are discussing toy models for origin of life, and the tensegrity might be another toy model we can look at.
Okay. Well, I really like this because they did something that was more like bone. I'm trying to find the words here, but it's different from your regular tissue. I'll look it up and maybe I'll send it to Bradley too, and it's an interesting paper.
Yeah.
All right, good.
Yeah, I'm interested in how you build a connectivity model for tensegrity. Like you're using like a mathematical matrix, and you're just putting in values for the different parts, or
COMSOL is just I have somebody at the door, but COMSOL builds it with geometry. Hello?
I think it's our dog this time.
Yeah. Well, hello, Morgan. How are you?
Did you get anywhere on the vehicles?
What was that?
Did you get anywhere on the vehicles?
Oh yeah, I'm going to present that in a minute here.
Oh, you are? Oh,
well,
okay.
I don't get anywhere, but I wanted to show the thing that I sent you in the
understanding.
Yeah.
Okay, so yeah. For my tensegrity model so far, all I've been doing is building the geometry.
Okay, yeah.
And I have a book on how to make tensegrity objects in MATLAB. And you have to put a half twist on them. They're like, you can't just have them like line up like this. You have to put a twist on them like this, and then they can be stable.
Oh, interesting.
But cells aren't like that. They're not like this, unless they're maybe a single-celled organism.
Well, I don't know. It looks like the top and bottom of an epithelium cell.
No, but they're lined up. They're not.
I understand. But maybe are the junctions lined up, or are they?
No, they look like they're lined up according.
Oh, I mean, are the top junctions lined up with the bottom junctions?
Yeah, that's the question.
That's the question, and I suspect nobody's asked that question because you'd have to You'd have to do
So you're
serial.
looking at somebody else's microscopy going, why didn't you just like take a picture over this way a little bit more?
Yeah, right. Maybe because, look, I can recall from the work of Beth Burnside, this would be 1960s maybe? I'm not sure. At any rate, she made pictures showing microtubules extending from the top to the bottom of epithelial cells.
Okay,
In the
I've got
cell.
that. I've
In
got
the cell.
that.
So maybe they connect junctions, top and bottom.
Well, yeah, they can.
They can, and therefore they might form your needed stability.
Well, what you do is you put a spring base on them, because that's the one paper I've been looking at.
Yeah.
And you attach the nose or the corners to a spring.
Yeah.
Base, and then the top part connects to each other. And so that's why I'm saying it's some sort of weird spider web, because the top is a spider web and the bottom is attached to a spring base.
Okay. But if the top and bottom are also attached, you might get a stable tensegrity structure.
Yeah.
If it's twisted.
Well, if it's twisted, maybe it's more stable, which would be a great experiment.
Yeah, maybe. And if the twist exists, one other thing, if the twist exists, it might be, there are two ways to twist, right? Left or right.
Yes.
Okay, and it could be that the left and right sides of a bilateral animal have opposite twists.
Well, there we go. How do I get to a microscopy at lab to take pictures of this?
You go. There's an electron microscope in the biology department.
Yes, there is.
Yeah, you can go just study.
So I should wander in there one day, and I'm going, I have this question.
Yeah. Hey, that's what. Did you know Chris Martin?
Yes, I went to his place when he was teaching in.
Well, see, he did electron microscopy of the top side of the cells, but he didn't bother with the bottom.
Yeah, well.
Okay, and he did it in botany.
No, well, botany's fine. I mean, that's another problem called
botany's
cells.
Botany's been absorbed into biology now.
Yeah, well, what I'm enthusiastic about is the entomology department of agriculture was absorbed into the biosystems department of engineering. I might be able to actually do some experiments with some flies. So look forward to it. Hopefully.
Okay.
Great. Yeah, that's great. I think that was a good conversation because it helps clarify, at least in my mind, what we're looking at when we talk about tensegrity and the kinds of things we need to model and the kinds of things we need data on, actually. I don't know if the data exists very, very commonly.
Bradley, are you familiar with the left-right asymmetry of bilateral animals?
Yeah.
Yeah, you actually can get different genes turned on left and right sides.
Right.
And the mechanism is, I'm not sure what it is. It could be mechanically based.
Yeah.
Okay.
Yeah. Okay.
See, if you take microtubules, if you think about the microtubules in a cell, the microtubules have a particular chirality. They all rotate the same way. So what this means, if the left and right sides of an animal are not bilaterally symmetric, because you can't get the microtubules on the left side are not a mirror image of the ones on the right side. They're all the same.
Right.
Okay, so if they do something mechanically different, because they're on the left and right sides, they might end up causing a change in gene expression.
Okay, that paper about the zebrafish by Merkel, that you had, saying that was the split between left-right asymmetry
Yeah.
a couple of months back. Maybe it was last winter.
I don't remember.
Describes some of that, but I think it was maybe poorly written because I read it several times and wasn't understanding how that worked. So you had it. Maybe I'll try to dig that up too.
Well, if the answer is tensegrity, don't expect it from a molecular biologist.
Oh, well, no, these people are physicists, so I have more confidence.
Yeah. Then they don't know their biology.
No. Well, I can be accused of that. I just took a cell biology course, cell cytoskeleton biology course on Coursera, just to see where my level of knowledge was.
Okay.
And I did fairly well with it, with just listening to the lectures once and answering the questions. So I do have a decent level of cell biology, I believe, after looking at that. And they were describing some of the biochemistry, and of course, I got so far with that, and then, well, no, I'm not understanding you. But they recommended I take a biochemistry course, and I'm going, don't know about that. But the paper you sent me about the cytoskeleton was using the same biochemistry and actin treadmilling forms as this course was describing. So it was very helpful because I was using this new knowledge of actin treadmilling to understand what the paper was saying, and it was nice.
Okay, good.
It's in my pile here somewhere, but yeah, thank you.
I forgot about it. Sorry.
Maybe I need to start making notes. Maybe like Bradley would like that.
They're called writing papers. I don't write notes.
They're what? Okay, let's see.
Yeah, a lot of this gets lost sometimes. There's just so much that. But I have to look back and see. I think I have like all the papers arranged in different weeks and things like that. So I'll take a look too.
Okay.
Why don't I move on to the thing I just promised that Dick asked me about the Braitenberg Vehicles, which we talked about several years ago in this group. In my other group, we're doing some work with it in terms of simulating the vehicles and their nervous system and all that. But this is a separate stream of work. This was something we talked about several years ago in terms of the morphology of the vehicles and how you can have these different parts and how you can analyze the shape and so Dick asked me, he's working on the origins of life, and I think this was the question was, can you do an inventory of parts on different vehicles? So to understand why that's relevant, you have to go back to the original source, which was Valentino Braitenberg's book on vehicles. And he publishes this in 1984. And the vehicles that the point of the book was to take, he's asking the question, how does the mind emerge? Or how does the mind evolve? And how can you go from like a bacteria to a simple nervous system that exhibits behaviors? So he uses this thought experiment where he built these vehicles. And the vehicles are, mechanical analogs of very simple organisms. So they could be like bacteria. They could be like they could be kind of like C. elegans, maybe with just one or two circuits. And so he designed these vehicles in the book He has about 8 of them, I think, with a couple of variants. So there may be about 12 total.
I think it was 14.
14, okay.
Yeah.
And so they all have their own shape. So like there's vehicle 1, which is a square. Let me share my screen. I can show this better. I'm just drawing from memory on this. I don't like. So vehicle 1, he did a typology of vehicles in the book. So he starts with the simplest one, which is 1, and that's like a square with 2 wheels, 2 light sensors at the front, and then connections between the sensors and effectors. I think there's like full connection there. So it's bilateral. So these are basically bilaterals, but they're vehicles, they're like little carts that move around in their environment. And these are the effectors of the wheels. This is what moves the organism. The sensors are in the front, which detect sensory information, and then it maps it back to the effectors. And the sensory information could be like light sources or chemical sources and that sort of thing. There are other vehicles, I can't remember, but there's some that, get really creative with their morphology. So it's something like this, where you have like multiple effectors, maybe like 4 wheels, and they may turn independently. A number of sensors would be like here. And then the nervous system would be more complex inside. He doesn't really specify. Like I guess he gives drawings, and that's about the end of the specification. But it's basically this type of thing where you go from simple to more complex, and he just draws these vehicles out, and roboticists have used this to test. They built robots where they build these little vehicles and physically, and they've wired them up and done experiments.
Yeah, you can buy one now.
Yeah.
It's called the Rumba vacuum cleaner.
Yeah. That's the basis for a lot of the sensor effector robotics.
Yeah, we had the first one. It didn't last very long. It didn't work well.
Yeah.
One of my sons bought one, and it does his whole house, and then it plugs itself in to recharge. And it even, when it plugs itself in, there's a vacuum cleaner in it, which takes all the dirt out of it.
Yeah. Those are really. Those look really great. I should get one for my place.
Oh yeah. The cost is $1,000.
Yeah.
Yeah. Somebody else can buy it for me. I need a gift.
It's a research item, so.
I'm going to post this thing. I can't find the.
It can't climb stairs, however.
Oh, yeah. Right.
You have to bring it up and downstairs.
This was Susan, a tug of war between filament treadmilling and myosin-induced contractility generates actin rings. Yeah,
it's an incomplete reference.
Okay,
but
should be able to find it. Okay, so that's the idea. But what I guess Dick wanted to know is, can we do an inventory of vehicles? In other words, can we take the parts of the vehicle and do an inventory? So the parts would be like the sensors, the effectors, the connections, parts of the body. So in this square instance, it's not so critical, but in something like this, you have a compound phenotype where you have the front, the snout maybe, and then this front part and the back part, and then have that as an inventory. And then
I guess in any sense is number 1 a subset of number 2.
Yeah, I mean, I guess the way he envisioned it was that you go from simple to complex. So you start with a motif like the square, and then you move up to, like, the square would be like here, and then you add on this part later. He doesn't get into like a lot of the sort of the phylogenetics of it, which I will talk about in a minute. But that's the way he envisions it. So it's very much just a thought experiment in that context. And I don't know if anyone's ever done any work on like talking about the sort of the inventory of parts or the phylogenetics. But I do have something that I worked on with the group, and then I put this on. This is a paper I put on something called the Winnower, which is like a open peer review thing. Yeah. So this is a paper that so I put together a couple years ago. This was in 2016, I think. And this was just describing, I did a talk in the group, and then I did this paper to put it together. So the idea is that you have Braitenberg vehicles, which are these vehicles, compound polygons, which are like taking these polygons, these shapes of the vehicles, and then composing them, and then evolutionary developmental structural complexity.
Oh, okay. Please. Bradley, could you send me a copy of that?
Yeah, I actually sent it to your email yesterday, so it's attached.
Oh, you did?
Yeah.
Okay, I'll look for it.
Okay. So the idea is that you have, and this paper kind of goes over like some comparisons of something called syllabic networks and other things. But this is basically, you have the Braitenberg vehicle, which is the composite polygon. You can use Kolmogorov complexity to evaluate it with respect to its physical complexity, how many sides it has, and that. And then the complexity metric, which is a coherence metric, so you can use a mathematical measure of complexity as well. So I'm going to skip over this first part, but I'm going to go to the second part, which I think is the more interesting thing here. This is the figure that I wanted. This is figure 3. And so the right side isn't the key part, but the left side is. So this is a phylogeny of a type 1 vehicle. And so this isn't so much the evolution of the body plan, but the evolution of the connectivity within the body. So you see in this case, I just took a picture out of Braitenberg's book, ABC, and then A has like this what they call ipsilateral connectivity to the left-hand side sensor to the left-hand side effector, and the same with the right side. Then B is like this contralateral connectivity, which is where you go from the right-hand sensor to the left-hand effector. And then the third variant is where you have both types of connectivity. And so you see that you can make a phylogeny out of that. But you could easily do this for like the morphology as well. So if you had a compound phenotype, you could have like where it has, you're adding on like parts of this rectangle. The first part of the rectangle on the bottom might be the sort of the basal condition. The second part, the front part of the rectangle might be the derived condition, which is what they say in phylogenetics. The basal would be like the adaptations that occur down at the base of the tree, and then the derived conditions are the ones that are on the branches. So the derived ones always come after the basal ones. So you're basically composing a phenotype based on these changes. So you have, to start with, every vehicle has wheels, and every vehicle has the square body. And the square body is, there's variation in the connectivity, but you could have, variation in that. Yeah.
Yeah, Bradley, the connecting lines could be regarded as parts in themselves.
Right.
Okay.
Yeah. Right. So this is the second part here, where we actually I have a couple of things labeled. So this is the same. I think it's the same tree, but it has the labeled changes. So at the bottom we have the rear wheels, long nose body, left and right frontal sensors. So those things are at the sort of the base of the tree. Then you have the hemispheric segregation of connectivity on the left. So this is where we say that that's derived. And there are ways you can do this. Cross connectivity is then derived between B and C, and then cross and hemispheric connectivity evolves from the cross connectivity in C. And so you can have this kind of phylogeny where, each trait has like an origin, a common ancestry, and then it moves forward. And like I said, you could do this with the morphology as well. Okay, so this is a syllabic network. This is just a way to characterize these different parts of the vehicle in terms of syllables. So this would be like right wheel, RW, LC would be left left connection, CC would be cross connection, FC would be forward connection or something like that. And then FSFS as another descriptor. And then it's just mapping this out into a network, all the parts of this, and then giving like a complexity measure. So you can use Kolmogorov complexity and Kolmogorov information. And so you're basically using a description with a length. And in this paper, I just used the sort of abbreviations. But you could use, like, if you're looking at DNA, you could use like a string of nucleotides. If you're looking at some other aspect of the vehicle, you could use a string of like a binary string, which might actually be useful, like looking at differentiation codes, which we've talked about, where the differentiation code is the location of the tissue in the differentiation tree, and then linking those together and saying, this is the complexity of this entire configuration here.
Okay.
Let's see if I can get, okay. So that's another syllabic network. And then this is actually some work on the geometry and evaluating it in terms of the connectivity of the different shapes that get composed together. So the idea is you should be able to draw lines through the edges and not do any self-crossings and then evaluate it that way. That's a topological analysis. And then there's this final part here, which I thought was interesting. We had, so this is figure 6. This kind of shows the differentiation tree, or it's a kind of an analog of a differentiation tree based on these compound polygons. So these would be like the regions here of the sort of this vehicle shape. And then you're sort of looking at this on 2 dimensions. You're looking at it as, you can't really see the labels here on this, but there are divisions in this direction and divisions in the sort of the horizontal direction and the vertical direction. And this comes from the square vehicle, the vehicle 1. And that square vehicle then is, decomposed into these sets of divisions, or, as this vehicle expands in size, what do we need? Do we need a number of divisions in one direction or another? And then those divisions can be analyzed as a differentiation tree, where these different parts are differentiating over time. So you have the smaller vehicle, you're maybe adding onto it as a composition, but you're just maybe replicating some part of some smaller part of the vehicle. It's like you get a polygon that replicates 3 times. That shows up. You can map that out in a differentiation tree, and then show maybe the smaller and larger components, or if a component shrinks or expands in some way as it composes a more complex phenotype, you can demonstrate that. And then that would all be kind of like in this origin of life thing, where you start with a very small primitive and you expand outward.
Okay, Bradley.
Yeah.
What I had in mind is, suppose you have a list of parts.
Yeah.
And suppose you take a sampling of the parts and connect them at random.
Okay.
What is the probability of getting a working vehicle?
Yeah. Well, yeah, you mean like if you had like a vehicle that was already complex, you took the parts. So it's kind of like a knockout experiment.
Take the part, yeah. Well, it's not simply a knockout. You throw them together at random.
Yeah.
Okay. And then the question is, what's the chance of getting a working vehicle?
Right.
And what I'm claiming is that if you do that enough times, you will get a working vehicle.
Right.
Okay, and that may be the origin of life in a way.
Yeah.
But the parts are thrown together, and sometimes they work.
Right.
And once they work, you're home free.
Yeah.
Okay, it's probably not that simple, but I've calculated the number of times you can throw them together is at least Avogadro's number.
Okay. Yeah.
And therefore, the probability that one's going to work is possibly quite high.
Yeah. Yeah. Especially if you have a lot of variance or a lot of replication.
Yeah, I'm not saying we have to do that many experiments, but if we do with a small system, the number of combinations of throwing them together is bearable. So you can do many experiments on a computer, and then you need a criterion for whether it works or not.
Right.
Okay. Like if the two wheels are in different places and they're going in opposite directions, they might just spin around.
Right.
Okay.
Yeah, and then of course we've created, in terms of connectivity, we've created this was something we just published on. It's a platform called BroadGenBrain, which actually just takes the internal topology of the brain of the vehicle and generates a bunch of variants from a genetic algorithm. And so in that case, you're actually creating different possibilities for like connectivity.
Right.
Some of them don't work, but some of them do work and get promoted to the next generation. So
yeah, that'll be a subset of this.
Yeah.
Throwing the mechanical frame also.
Yeah.
But that's why I asked Susan, that's why I asked about Tensegrity. If you throw the Tensegrity parts together at random, what's the chance of getting a Tensegrity structure that doesn't collapse?
Yeah.
Yeah. If you've got it supported, maybe it's like I said, like a spider web, and it works just because it's connected at the edge.
Okay.
It's on its own and has to have a twist.
Yeah.
Okay.
I put the book by Gann here. It's you can get it for, I think you can get it for free just online.
Okay.
And computational modeling of tensegrity structures, and that's where they show you that you need to put this half twist on them. Even a triangular one has to have a twist, and other configurations. So it was a somewhat difficult read. I read half of it and that was fine, and the last half of the book is a bit difficult.
Yeah.
Just put it that way. And then there's an MIT course online about origami and tensegrity. And I wanted to take it, but it said I needed to know my differential equations. So I've started off relearning my differential equations.
Oh boy.
With the course.
It's been a long time.
I need to know it anyway. Somebody's going to think I know that, and they're going to throw something at me. And I don't want to look like a deer in headlights, because that's a crash. Yeah.
Yeah. Well, thanks.
Also MIT online.
Oh yeah.
Through Coursera, actually.
Yeah. Well, yeah. Thanks for these references, Susan. That's interesting stuff.
Yeah, and I still haven't found my most favorite paper. I better locate it. I have it written down and quoted, but I reorganized myself, so now I lost my papers. I even lost my book on optical coherence elastography.
Oh, wow.
This is not.
Back up.
So I wanted to go over a paper. Dick mentioned in the meeting that we had there might be interactions between physical forces and molecular forces in some cases of morphogenesis, particularly this tensegrity structure. So in tensegrity, it's dynamic tension. It's mediated by different parts of the phenotype being joined together in different ways. Molecular pathways may either affect that or be triggered by that and affect it subsequent to its configuration. So this is all fascinating, but I wanted to go over a paper that it was recently, let's share my screen, recently came out. I think this is in Nature. So this paper is bending force as a nucleotide state, jointly regulate F-actin structure. So this is about the cytoskeleton and some of the actin components in that structure and how molecular factors play a role in that or are jointly regulated. So let's read the abstract. So APT hydrolysis coupled actin polymerization, which is the creation of actin molecules or the polymerization of actin molecules using ATP hydrolysis. It's actually coupled as a process, is a fundamental mechanism of cellular force generation. So this is where cells move around, they're motile. And to be motile, they don't use muscles per se, they use actin molecules. And of course, actin molecules are components of muscle, the muscles that we have in our body. So they behave in a similar way. They're contractile, and so when you expand and contract them, they generate forces and they generate movement. So in turn, so this is the first part of this, the first sentence. So cellular force generation, just so we know what we're talking about. In turn, force an actin filament, or F-actin, so you can think of F-actin as filament actin, a nucleotide state, regulate actin dynamics by tuning an F-actin's engagement of actin-binding proteins. They're mechanisms that are unclear. So what we have is we have force, which is a physical property. We can measure force. An actin filament nucleotide state, or F-actin nucleotide state, regulate these actin dynamics. And so they're mechanisms that involve actin-binding proteins and this whole process of polymerizing actin so that the cell can move. Here we show that the nucleotide state of actin modulates F-actin structural transitions evoked by bending forces. So a lot of different types of forces are generated as the cell is moving around. One of those are bending forces, which actually the nucleotide state of actin modulates F-actin structural transitions. So there's a modulation that goes on through bending, the action of bending forces. And this affects, of course, nucleotide state. Crayon electron microscopy structures of ADP F-actin and ADP PI F-actin with sufficient resolution to visualize bound solvent reveal inter-subunit interfaces bridged by water molecules that could mediate filament lattice flexibility. Despite extensive ordered solvent differences in the nucleotide cleft, these structures feature nearly identical lattices and essentially indistinguishable protein backbone conformations, which is the shape of the protein backbone, that are unlikely to be discriminable by actin-binding proteins. We next introduce a machine learning-enabled pipeline for reconstructing bent filaments, enabling us to visualize both continuous structural variability and side chain level detail. So this is going to be a lot of imaging of these structures and proteins, and then some analysis of the protein structure as well. Bent F-actin structures reveal rearrangements at inter-subunit interfaces characterized by substantial alterations of helical twists and deformations in individual protomers, transitions that are distinct in ADP F-actin and ADP PI F-actin. So this is what we were talking about a little bit earlier about the left-right differences in the structure of the cytoskeleton and different helical twists that are necessary for tensegrity. But in this case, we're talking about alterations of the helical twist in some of these proteins. So you can see that you have structure at multiple scales in cells that are important for function. This suggests that the phosphate rigidifies actin subunits to alter the bending structural landscape of F-actin. So the bending structural landscape changes as bending forces are introduced, and it's a feedback loop, and you get increased structure that sort of favors bending forces as bending forces are introduced. As bending forces evoke nucleotide state-dependent conformational transitions of sufficient magnitude to be detected by actin-binding proteins, we propose that actin nucleotide state can serve as a co-regulator of F-actin mechanical regulation. So that's the abstract. Let's see if we can find some images. Here are some nice images of the nucleotide cleft water networks. They're remodeled upon phosphate released by F-actin. In A, we have cryo-EM maps of this ADM F-actin, that's on the left, and the ADM PI. So we talked about those two things in the abstract. This is their structure using cryo-electron microscopy. In B, atomic models of the densities are shown. There's the barbed end, which is BE. And I don't know if they actually show, okay, they show it in here somewhere. But basically you can see these different aspects of the structure. They're using these atomic models, and they show they're color-coded. So atomic models of the ADP F-actin in blue here, and the ADP PI F-actin clefts in orange. So you can see this. And so this is just information about the structure. And then C is superposition of individual ADP F-actin blue and ADP PI F-actin protomers, blue and orange respectively. So they just place them on top of one another, and they show how this is the nuclear cleft, this is the D loop. Very, very advanced structural biology here. Then we get into this water molecule aspect. So the water molecules mediate key longitudinal and lateral contacts in F-actin. So this, at the top, you have your water molecules, violet, contained within the filament core. So you can see the violet dots here that are within this filament core. And then B talks about solvent-mediated contacts at lateral and longitudinal interfaces. So the lateral are at the top, the longitudinal are at the bottom. These are the interfaces of ADP actin, which are in shades of blue, and ADP PI F-actin, which are shades of orange. So you can see these two. This is P and this is PI, and you can see these differences in the structure here. This figure shows cryo-electron microscopy reconstructions of mechanically deformed F-actin reveal bend-twist coupling. And so this is where we get into the forces and the movement. You have this bend-twist coupling here. And in D especially, you see these stitched volumes of straight and bent maps. And you can see this for how this sort of unfolds. In this case, actin nucleotide state modulates subunit sharing during filament bending. So this shows kind of the action of how subunits are shared during this bending force. So here we have the bending. This figure actually shows it, and you see that the cartoon shows the bending and how this shears off and changes the structure. And this is in the ADP. This is in the ADP PI, where the bending has a little bit different action. Presence of the PI in the nucleotide cleft stiffens the subunit and alters its deformation landscape. So this PI being in this cleft. So each of these have a cleft, and in ADP, plain ADP, there's nothing in the cleft. The bending sort of, this thing collapses and it changes the conformational structure here. The steric incursion of neighbors deforms the subunit. So the subunit gets deformed. This in ADP PI, where you have these PIs occupying the cleft, they stiffen the subunit and alters the deformation landscape, so this doesn't change as much. And so that's that's all they have in terms of figures. So I hope going over this paper was useful in thinking about some of the things we've talked about in the meeting. I hope you learned something. All right. Well, that's all I had for today. Did you have anything else you wanted to?
Okay. Well, that's great, actually. So if we can get an inventory of parts, let's think about throwing them together at random with random connections and a criterion for whether or not the vehicle works.
Yeah.
Okay.
Yeah.
In the case of the tensegrity, the question is, do you get a stable tensegrity structure, or does it collapse?
Yeah, and if you put it in COMSOL and it collapses, it just says empty matrices or eigenvalues are off scale. Solution does not converge.
Okay. Well,
solution does not converge. I'm beginning to hate that phrase.
Excuse me.
The way I get these large numbers is there's a hypothesis that vesicles come from amphiphiles from meteorites. Okay? And in order to get vesicles, you have to have what's called the CVC, the critical vesicle concentration. So you have to concentrate these things, these molecules, and then they will form vesicles. Okay? So what I did is I looked at, what do I call it? I looked at lakes that do not drain to the ocean. Okay, so any rivers flowing into them keep building up the concentration of whatever's in the rivers. Okay, and I calculated that you should be able to get a 10 to the 40th vesicles and get them in under 100,000 years. And then the vesicles could each one be an experiment in that molecules that go inside the vesicles can work together and possibly produce a living organism. Okay?
Yeah.
So that's the basic idea. And when you start getting numbers like 10 to the 40th, it becomes plausible. Okay? It's hard to do the experiment, but even in a computer, you can't test 10 to the 40th item, so the reasoning seems plausible. Okay? So that's the basic idea. So that's why I was thinking a toy model might be a good idea, because with a toy model we can play with it without worrying about simulating metabolism itself.
Yeah.
Because the metabolism would be the interactions between whatever molecules end up in these vesicles, and you'd have to simulate each one and have a criterion for whether or not it's a viable metabolism. But in our case, if we do a toy model, the criterion for something being working, it doesn't have to be alive, but if it meets the criteria, then you can get an idea of whether or not this approach works, which I think it will work, but it's just a question of how often.
Yeah.
The other thing that's happening in the literature is if you make random peptides, throw amino acids together in random sequences, occasionally you do get a peptide that is catalytic and can run a particular reaction. So the idea is not completely new, but it's been demonstrated for peptides, and I think for RNAs also.
Yeah.
Okay, Morgan put some software now.
Yeah.
Well, so hi there. Sorry, still kind of reeling with COVID here, but there's some good software that again more in the kind of Braitenberg Vehicles approach, in the sense of these are kind of
Tensegrity robotics, yeah.
I mean they're and they're not console's like a full featured, finite element environment, right? So, the kind of the burdens on you to create the appropriate geometric objects and things like that. And, just to even get started, these are kind of more limited approaches, but again, they were very much designed to create these simple structures that might be useful, or certainly. But this is unfortunately kind of an old NASA project. Like I forget the guy's name. It's kind of an unusual name. I can't remember the lead PI's. Anyway, it should be there, and I believe he's still trying to work on something similar, although he's no longer at NASA. But
the idea is to build robots that can move over a rough terrain. Correct.
Yes, as well as that they can get damaged and still be mobile, right?
Yeah.
So there's a kind of resilience engineering in this approach that they thought would be super useful in kind of Mars missions and things where something could go wrong, but it was really interesting, right? Because you don't think about that with a car, right? Like a car breaks down, then you just go get a spare part, kind of thing.
Yeah.
Right? And you take it to the shop.
Not out here.
Yeah.
Car breaks down, you get a farmer.
But very, very interested following the conversation and again, like hopefully this week I'll with the big NerdTechX hackathon done, I can focus on some other things, and I think there are some other good software projects that try to get at these kind of more cellular models.
Yeah. Okay, well, there's an interesting prospect. See, if you did a Tensegrity robot, the criterion could be as to how far can it move.
Yeah. Well, and that was, again, this was like a big part of his work, right? was making I hesitate to call it a soft body robot, but in a sense, I mean, it's a strange mechanical design, though, right? Because he wanted to let it, for you to kind of be able to lose tension and then reconfigure into another.
Reconfigure too.
Yeah. Well, that was like, if something goes wrong, it should be able to. One, you can just imagine it in the sense of like dropping the robot from a height, and this would allow it to kind of bounce and absorb. Two, that can also be seen as like a possibility of reconfiguring to allow the kind of the body mechanics to have a different system of body mechanics.
Yeah, that goes one step further.
Yeah.
I was just thinking of throwing things together, but then you'd have a static thing. What you're saying?
Yeah.
One step further and have it self-repair.
Well, but I like that in the sense of like to me that again, and thinking about I'm trying to think of the particular evolutionary biologist, but when you think about evolution, not as survival of the fittest, but as the most adaptable, right? That these kinds of resilient designs and these possible reconfigurations make a lot more sense. And even though yes, at the origin of life, you kind of need them to come together at some point, but this would be the kind of the come together that then permits kind of future reconfigurations. and it's very interesting. And again, so I definitely recommend. I'll try and dig in and find more references today.
Okay, well, I have a paper about tensegrity repair.
Oh, really?
Yeah, I do somewhere. And I'll try to get that as well.
Okay, good. Okay.
Yeah, but I did think it fit in nicely with Bradley's Braitenberg Vehicles kind of overview today. That this NASA AIM stuff is kind of perfect for.
Yeah, Bradley, we're I guess none of the vehicles were self-repairing, were they?
No, well, yeah, I mean, Braitenberg didn't think of it that way, really. He just kind of said, this is a vehicle you can build. And he didn't talk about, like, how it gets organized, or how you might have an origin of it, because that's actually kind of important, though, because if you think about biology development, you have these things getting put together, and then you have regenerative processes.
The problem of origin of life is how do you get from no structure to a structure that works?
Right.
Did you see, Bradley, the Michael Levin retweeted a video of an origin of multicellularity talk?
Yeah, I think so.
I didn't. Do you know this Georgia Tech guy? It's like Will something.
Yeah, I think so. I can't remember. Will. Yeah, I think I know who that is.
Okay.
He works on yeast or something like that.
That's right. Yeah. Exactly. But I don't. Yeah. But I want to see what that this particular Ames guy is doing now. I think he left NASA like 10 or 12 years ago.
Interesting stuff.
All right.
I'll look for some of these papers too. So,
okay,
but I gave you that nice one by Gann that actually shows you the twist that it needs.
Yeah. All right. Well, thanks for meeting, everyone. We'll keep talking about this, I guess. Follow up on it.
Good session.
Yeah, good session.
Oh, did you get to talk to Nora a bit?
Yeah, a little bit. Yeah, I did.
Okay, good. Okay. Yeah, I shouldn't always be there.
Yeah, I know.
Okay.
All right.
Okay, good. Have a good day.
Have a good week.
Take care.
Thank you.
Bye.
Bye.